So the title of my talk is Pan-Genome Analysis, Revealed Gene Loss During Tomato Domestication, and also uncovers a real allele that regulates food flavor. So in 2012, so tomato reference genome was published. So that at that time is a huge event at BTI. So, but also it's a huge event for the consortium. So the paper was published in Nature, involved like I think more than 10 other countries. The number of authors on this paper is more than 300. More than 300 authors on this paper. And after the publication of this paper, I mean, the genome was heavily used. And now it's cited close to like 2,000 times, just in like 7 years. But later we realized that one reference genome is not good enough. And you can see from this figure, I'm sorry. You can see from this figure that tomato, even only the cultivated tomato, they have very wide phenotypic variance. You can see the different color, different shape, different size, and also the taste different. And they also have even higher metabolic diversity even than the cultivated species, the wild species. So this implies that some unique genes exist in some certain accessions. So that's not captured by the reference genome, but those genes are very important because they contribute to unique traits of a certain accession. And also the wild species, they may contain genes that are absent in cultivated tomato. And this wild relatives has been commonly used as a source for beneficial traits like disease resistance. And a lot of disease-resistant genes that are not existing in cultivated tomato. A very good example is the PTO gene that cloned by Greg Martin's group. They are not in plants. They are in Pimpilifolium in this accession, in this species. So that's the reason that triggered us to do a whole survey of the pan-genome. So next, what is the pan-genome? So the pan-genome concept was developed in 2005. As first developed in bacteria genome analysis. So in this paper, they compared 6 bacteria genomes, and then they coined this concept called pan-genome. And of course, the obvious explanation of pan-genome is that you have a reference, then the reference genome, right? Then you have non-reference gene, and together become a pan-genome. But more importantly, they developed this concept, the core genome and the dispensable genome. The core genome is, the implant people mainly focus on genes, not the genome, the entire genome region. So I'm mainly talking about the concept in the plant field. So the core genome is genes shared by all organisms. So like in tomato, if you sequence like 1,000 accessions, you would find like some genes, they exist in all 1,000 accessions. So those are core genes, that are essential genes. But also you have dispensable genome. The genes are found in 1 to n-1 organisms. So those are genes that are contribute to the unique traits of the organism. So this, both are important, but these are kind of, even more interesting because they related to some interesting traits. So that's why, we collect data from the public domain and try to build a pan-genome. So this is a tree, it's not complete, but just give you an idea. In tomato, we have a lycopodium clay. People also call this a red clay. So these fruits are red. And they have the cultivated one, the lycopodium SLL. And also ceratiforme, that's a landrace. It's also called cherry tomato. And we have Chismania and Galacpancy. These two are highly related to carpet species, but they are never used for domestication. It's just one species, never used. And this Pembelifolium is a wild progenitor, the direct wild progenitor of Cardiff tomato. So the Cardiff tomato was domesticated from Pembelifolium. And also we have some green clay. That's a wild species, the fruit is green. And some of them are purple, some of them, but I mean, in general, most of them are green. So we want to build a plant genome. In this study, we just focus on this clay, like a Poscan clay, because their genome are very close. And also it's much easier to work. If you include like Panalia, although another very popular species. But if you include Panalia, it's very challenging to build a plant genome, because the Panalia genome size is even more than 200 bigger than Cardiff species. It's very difficult to integrate. But I mean, in the future, I think people will find a way. Even if it's not my group, other group will find a way to integrate this. So after the publication of the tomato genome, there's quite a few studies. They do genome resequencing. They are trying to understand the evolution, the variants, identify the variation map that try to associate with interesting traits. And we collect those data from public domain. And also we also sequenced 166 accessions for this project and the tomato Waratone project. And Lucas and me are involved in this project. This project was led by Esther Vandernet from University of Georgia. And in total, we got 725 tomato accessions. And in this clay, in Lac-Poscan clay. I mean, mainly about half of them are Cardiff species. And 265 are Solatiformi, that's a landrace. And we got 78 Pembelifolium, but we only got 8 from these two. But I mean, these two is just include, because they're close related, they are not that important in domestication or breeding. So these are short read sequence. Some of them, the coverage is very low, but still we want to do a de novo assembly for each of them. So for this 725, we did a de novo assembly, and I mean, as you can expect, for the short reads and the low coverage, you will not get a good high quality assembly. And you can see the effective length is very short. The canonical length is very short. But in the end, we got like 4 gig combined contiguous. And we still keep this contiguous because this contiguous contains a lot of information. And if recently someone asked me to ask us to extract one gene sequence, the gene sequence from all these accessions for one gene, then you can use this to extract the sequence. But in the plant genome, we first remove the redundancy because this contains a lot of redundancy. And then we also remove the contaminations from chloroplast, from bacteria, from virus. And in the end, we got like 351 million base pair non-redundant, non-contaminated sequence from this plant genome. And then we combine with this tomato reference genome, and we got like 1.179 gig base plant genome. So the plant genome size was increased by about like maybe 40%. And we did annotation on this plant genome and the reference and the non-reference. This is the comparison of the reference and non-reference on the repeated development. You can clearly see the non-reference genome contains much higher repeated elements. And that's also one reason that it was not assembled in the reference genome. So we then did a gene prediction for this non-reference genome. As Paul already mentioned, we got a total of 4,873 genes that was annotated in the non-reference genome. And for reference genome, we also did a high-depth genome resequencing. So the reference was HANS. 1706. We did 45x sequencing. We used it as a control, and we aligned this reads to this reference genome. And surprisingly, we found that a total of 272 genes from this annotation, this coverage is very low. The gene was not, it is not covered. Or there, I mean, there's 240 of them, there's no read cover at all. And then we checked these genes. We found like almost all of them are on chromosome 0. That's I mean, not a real chromosome, it's a feature chromosome. We just connect them. And then we checked the annotation of these 272 genes. We found them they're not from tomato. They're from bacteria, they're from fungi, they're from other plants. So we are highly confident that these 272 genes are contaminations in the reference genome. But that's not unexpected, because I mean almost all the genome sequencing project, it's very hard to clean out everything. You expect some level of contamination. But this detailed analysis, I mean, can confidently remove this 272 genes from this reference genome. So then, in total, we got like 40,369 genes in the entire plant genome. And we found that there are several genes, well-known genes, that previously well-characterized genes, that are actually in the Hans genome. They covered by Hans read, but it was not assembled. So that includes the game A and the pin2 gene. These are very well-characterized genes, but that's not assembled in the genome. That's also understandable, because in the genome sequence you can't assemble the entire genome. I mean, most of the gene space was assembled. Normally it's like 96, 97%, but you still got 1-2% or 2-3% was missing in the genome. That's normal. But in the pangenome analysis, we can capture some of them. And also, we found some well-characterized genes that are not present in Hans genome, but they captured in the pangenome. The one I mentioned before is the PTO gene. Mainly, we found several examples, but this is a very good example for disease-resistant genes. They are not in the cultivar species, but they are in the Pimpelifolium, like PTO. And we checked the expression of reference and non-reference genes. This is another group in China. They did RNA-seq on orange stage fruit. They sequenced about 400 accessions on this stage, and we checked the expression distribution. We found that expression of reference gene was much higher than non-reference genes. And later, maybe I can tell you why. And after we got this pangenome, I mean, we wanted to check the presence and the absence pattern, the variation pattern in the entire population. So due to the limit of the data, some data set had very low coverage, even like 2x, 3x coverage. So those data was not good for peer review call, because some peer review call using that data could be due to the low coverage is not real absent in that reference, in that accession. It's due to the sequence did not capture that part. So we did quite an extensive analysis, and we later found that we used depths greater than 4x, but not only depths greater than 4x, we're also using that the reads cover like more than 600 million base pair of the genome. And we did analysis, used all these accessions. We found using these accessions, you will capture more than 99% of the genes in the pangenome. So we have less than 1% error rate. So that's why we use this 586 accessions out of that 712 for this analysis. We identified PVs, and we analyzed the model of the PV pattern, and we found the genome, the pangenome is closed. So what it means is closed? That means that if you add another accession to this pangenome, the total number will not increase much. maybe increase 1, 2, or 10. But it's kind of like the saturated. And also the core genome is also kind of like closed. And that means if you add another accession, the core genome will not be decreased much. So that means the accession we use in the lactopostine clay, they captured probably the majority of the genes, if not all of the genes. So then we divide, according to previous studies, we divide these genes into 4 different categories. The core, the soft core, a lot of people do not like this term, the soft core. A lot of people mention to me when I give talk, why I use this term? To be clear, this is a term not invented by me. It's used in the first initial pangenome studies. We have to speak with it. So we have core genome, core genes, soft core, shell, and cloud. The core genes, that means they present in all the accessions, 100%. The soft core, in our study, we use 99 greater than 99%, but less than 1%, less than 100%. And we have shell genes, that's 1% to 99%. They present in the 1% to 99% of the accessions. Then cloud genes, these genes are quite unique. They only present in less than 1% of the accessions. We further group them into highly conserved genes and flexible genes. We checked the flexible genes. We did a gluten enrichment analysis, and we found most of them are related to defensive response. That's what we expected, because during domestication, there's a lot of defensive response gene was lost. And one unique feature of tomato pangenome is that the core gene occupies a very high percent of in the pangenome. It's 74%, and it's higher than Rhodopsis, brassica, wheat, rice, white soybean, and brachypodium. So it's higher than several previous pangenome building other crops. And I think this implies that the tomato has very narrow genetic diversity. That's well known. That's the one reason you got the high percentage of the core genes, the genes present in all the accessions. And we checked the expression of these different groups. We found that the conserved gene has much higher expression than flexible genes, because that's only from one stage. That's understandable because most of the genes that are not involved in this stage, those flexible genes, that contribute to different traits. And for conserved genes like reference, non-reference, there's no big difference. But for flexible genes, reference and non-reference, they have quite a lot of difference. So as I mentioned before, tomato, the cultivated tomato was domesticated directly from Pembedifolium, from the wild species Pembedifolium. Pembedifolium is very small, and you can see the size difference. And now the size, the cultivated tomato, the size is about 100 times of the wild species. And during this breeding process, people further have the domesticated Pembedifolium to start forming. So this process is called domestication. Then the modern breeding program, they did some improvement. They improved the Soratformi to the very large, the big, like a Tuscan cultivated tomato. And this process is called improvement. So next we want to see the PAV pattern of those genes in the pangenome. And I want to see how the pattern changes during domestication and improvement. And first we checked the gene number changes. And we found that at the Pembedifolium, this is a Soratformi, this is a cultivated tomato. You can clearly see this gene loss. And it is very significant from Pembaliform to Ceratiformi, then from Ceratiformi to Lycoposcan. You can clearly see a gene number reduction. That means that quite a lot of genes was lost during domestication and also during improvement. And we then use this PAV pattern. This kind of PAV present absent variation is similar to SMP, right? SMP is a single nucleotide polymorphism. This is a single gene polymorphism, is the PAV. So we use this PAV to construct phylogenetic tree, and also did a population structure analysis. And we found that we can, for the Pembaliform clay species, this accessions can be divided into two subclade. One is SP1 and one is SP2. So this SP2 is very kind of interesting because this includes accessions from the coastal region of northern Ecuador. And this clay also have much fewer genes than this main clay SP1. So this implies probably a major bottleneck when these accessions they migrate to the northern mountain area. There's a gene loss. They probably adapt to this specific environment. Then we can see from SP1 to Ecuador, this Ceratiformi, then Peru Ceratiformi, and Mexican Ceratiformi, and this continuing decrease. And this was consistent with the domestication trajectory that people proposed. And also, this could be the direct ancestor of the cultivar tomato. The domestication and the improvement mainly happened in Mexico. And one interesting thing we found is that this album has much less genes than modern cultivar species, that the F1 hybrid and also the inbred line. So that means, there's some new genes. People started to realize some interesting traits in those wild species or in the Ceratiformi. Then they try to bring those genes back. And you can clearly see that this F1 or modern cultivar, they have much higher gene content. And that's also the case for the flavor gene I will mention later. So we then investigated the gene selection during tomato domestication and improvement, and we did some comparison, the gene frequency comparison, present absent frequency comparison between the Ceratiformi and the Pembaliform. These genes are related to domestication. And we also compared Ceratiformi with cultivar tomato, like Poscan. This gene identified in this comparison was related to improvement. And interestingly, we found that most genes we caught unfavorable because their frequency is highly decreased in cultivar tomato. And more than 1,000 genes, like 1,200 genes, their frequency was much lower in Ceratiformi than Pembaliform. Only 100, the frequency was high. This was consistent in improvement stage. You can see 800 was higher, only 12. 800 was low, only 12 was higher in cultivar tomato, in Lycoposcan. This is also an indication of gene loss or negative selection during this breeding history. And another interesting we find that if a gene was selected against during domestication, it's also selected against during improvement. There's a continuous selection during breeding and the improvement. And only like 41 genes, and this red genes means continuous decrease, the green means continuous increase. Only 41 out of this like 1,300 genes was showing different pattern. In this one was increased, in that one was decreased. But I mean, more than 90% of them, they have shown continuous pattern for both. I mean, for both domestication-related genes or improvement-associated genes. We checked the domestication selected genes and did the GOTOM analysis. Also, there are many involved in defense response, and some of them related to aging, senescence. For the improvement, it's same. We got a defense response. And we did not find enriched gene families in favorable genes during improvement. The first is the improvement, we only got very few genes. And also, during improvement and different accessions, people, they improve different traits in different accessions. Not like a single trait, not like fruit size. I mean, fruit size, people know that they increase in all the accessions. But for some unique traits, they just improved in one certain accessions. So that's why we do GOTOM enrichment analysis, they will not show up. And next, we did the promoter analysis because we know that some genes was not selected just by presence and absence. Some genes was selected by the regulatory sequence, the regulatory expression pattern. We also found the similar stuff with the genes, and most of them are consistently up or down during the both process, domestication and improvement. And very interestingly, from this selected promoter region, we identified one specific promoter. This promoter is a POA that's about 4,000 base pairs. And this is in the promoter region of Tamalock C. The Tamalock C encodes a LOX enzyme, and they can initiate the metabolism of fatty acids into C5 and C6 volatiles. And it's associated with tomato fruit flavor. And this promoter, this 4,000 base pair promoter was present in 86% of the pimeriform accession, but highly decreased in ceratoforming in the lanolase, and then continued decrease in lycopodium accessions. And this indicated a strong negative selection during domestication and improvement. And we also found the presence of this promoter was strongly positively correlate with the expression of Tamalock C. If you have this promoter, the expression is much, much higher than you don't have this promoter. And we did a further analysis on this promoter region. We found that actually this is not a simple present-absent variation. It's a substitution. So there are two alleles of this promoter in this region. One is this green one is a non-reference allele. That's the one we identified. And the orange one, that's a reference allele that's already present in the reference genome. Then we check the distribution of this promoter, these two promoter alleles, and we found that the number of accessions that contain homologous reference allele, the number is very small in pimeriform, but in alone, most of them contain the homologous reference allele. I mean, 2 copies of the allele are from the reference. And we only identified 6 accessions that contain homozygous non-reference allele. Only have these 6 accessions, and 2 from pimeriform, 4 from ceratoform, and none from modern varieties. They are homozygous non-reference allele. But we identified a lot that they contain both alleles. That means they're hybrid alleles in this region. They're mostly in Pamplifolium. And we also identified some in Ceratophrumi and identified some from Elm and modern cardiac. And if you check this non-reference allele frequency, you can see this consistent with the number of the accessions containing this allele that decrease throughout the domestication. But one interesting thing is that in modern cardiac, this non-reference allele was brought back. You can see the increase of this non-reference allele. The people started to bring back this allele, but we don't know why. I mean, probably people start to focus on the flavor, or this one may relate to some disease resistance or something. But this is a fact that the modern cultivar they contain much higher allele frequency of this non-reference allele. So we are, for this accession, the RA2093, we identified how many I have? Okay, I go fast. So we did not see any structure changes, gene structure change in this gene. So that means this was mainly controlled by this promoter region. And for this RA2093, we have a population, a real population, that's the parents of one of the real populations. One is RA23, one is another cultivar. And this cultivar contains the homozygous reference allele. And the RA2093 contains the homozygous non-reference allele. So we have a rare population, and we check we check the expression pattern. I'm sorry, I'm in a hurry. So this one, we did the expression we checked the expression of those in the natural population, not in this real population. We found the accessions containing the hybrid alleles has much higher expression than contains both the non-reference, homozygous non-reference, and homozygous reference alleles. And also in these two accessions, we see the different expression pattern of this TMLOC3 gene. And we did promoter analysis. We did find the promoter contains some unique binding site that may contribute to their differential expression. As I mentioned, we have a recombinant inverse line, and we did RNA-seq for all these lines. We co-SNP and co-expression for this line, and we did the QTL mapping on volunteers. In total, we mapped 65 volunteers, and we found 29 of them shared 149 region on chromosome 1 that contain TMLOC1. And this region only have 6 genes, and only this gene was actively expressed in ripe fruit, and only this gene has homozygous structure variation in this one. So through this QTL mapping, we can easily narrow down to this gene. And for all 90, I will skip this one. And as expected, 19 fatty acid derived volunteers, they are mapped to this region because we know this function. The catalyzed fatty acid to C5 and C4 volunteers. But it was unexpected that none out of these 11 apocarotenoids, they also map to this region. I mean, this also suggests that TMLOC3 may contribute to the biosynthesis of apocarotenoids. And then we did, actually Jim's lab did this transgenic analysis. We knocked down this TMLOC3 gene, and we found the expression of this another gene, CCD1, that's known to be involved in this apocarotenoid biosynthesis. And we can see that this one was not changed. The expression of that gene was not changed. But the expression of this TMLOC3 in antisense was highly decreased. And we found all these 9 apocarotenoids mapped to that region was decreased. And the 2 was not mapped to that region, that they did not show changes. And we also did this, confirmed this in arthropsis. We found the arthropsis homolog genes, AT LOX2, and we did the transgenic plant with the reduced expression, And we also find a mutant that carry a point mutation causing premature stop codon. And also showing in this transgenic plants, they are decreased expression, decreased content of those opercarinoids. So this confirms that opercarinoids, the tamaloxy was involved in opercarinoid production. So I will give a brief summary. So we built a pangenome for tomato, and with a total number of genes is 40,369, and of which 4,873 were absent from the reference genome. I mean, not from the reference, it's from the reference genome. Some of them are in the genome but not assembled. Around 18% of the pan genes displayed a PAV behavior. That means they have present and absent variations in the accessions. And some of them are predicted to be involved in important biological process. So gene reduction and loss is the major trend during domestication and subsequent improvement. And a large portion of defense response related genes tend to be lost. Domestication and improvement have a similar gene selection preference. They either can use to select against or either select for. So more than 1,000 genes have been negative selected to human domestication and subsequent improvement. So we identified a rare allele was detected in the pan genome, which regulates the expression of tamaloxy, then which further affected the tomato flavor. And a novel role of tamaloxy involved in the opercarinoid production was confirmed. So now we reported this pan genome, then what's next? So these are several ideas we have now. So recently, a much improved tomato reference genome was released. Actually, last week was released. So we plan to update the pan genome based on this new reference genome. This reference genome was assembled using PacBio, using BioNano, using HiC, much more accurate. And also, there's some activity going on to plan to improve pan genome with de novo assembled reference level genomes. So the genome we assembled is not reference level, it's from short reads. And also last week, Zach Lippmann and Michael Schwartz group, they released 13 tomato genomes that are assembled using nanopore, and they are reference level. And if you integrate them, then this pan genome quality would be much higher. There's another consortium led by David Francis from Ohio State, and Sam Hilton and Tom Lee from University of Florida. They're using this NR gene technology to do de novo assembly non-tomato genomes. 7 are cultivars, 1 is from Camelifolium, and 1 is from Habachitis. So this genome would be a great resource to expand the pan genome. And also, I mean, with the future pan genome maybe focused on those clades, on the green fruit, because green fruit, they contain much more genes and useful genes that can be used to improve the cultured tomato, and also the not present in cultured tomato. So with that, I thank my postdoc Lei, who is in the audience, who did most of the bioinformatics work, and Itay from Jim's lab, a previous postdoc in Jim's lab, now a PI in Israel, who did the characterization of the tamaloxy allele and function. And Heracles and Danish team provided the metabolite data for QTL mapping, and someone at Astor, they generated those sequences. And I also thank all other co-authors. I can't mention each of them. All other co-authors in this paper, and thank you. I would like to take any questions.
So we should have time for a couple questions.
Yes.
So 4,800 new genes seems like a lot of new genes. And I would imagine that a lot of the exceptions would have pretty distinct alleles from the reference genome. So I'm just curious, how do you define, what is the defining characteristic between a different allele and a new gene?
That's a very good question. Yeah, that's a good question. Also asking about the review. And actually, we did a lot of analysis. We check. We are trying to differentiate the alleles and the new genes, right? But for alleles, normally you have very high sequence similarity. And in our study, we use 90%. And we did the different cutoff. We start from 70%, then 80%, I mean each 70, 72, 74, to 100. We found at the beginning it's increase. I mean it's a linear or it's kind of like linear. It's a increase. I forgot the figure. I mean at 90%, no. At the beginning it's become flat. It's stable because using different rows they can distinguish different genes. But if you go above 90%, the novel genes will be substantially increased. That means those increases are not novel genes, that could be alleles. So in our study, and this analysis was also done in bacteria, the original, the pioneer bacterial pendulum studies. We did this analysis, we found 90% is a reasonable cutoff. But I'm sure that some of them could be alleles, but I think the chance is very, very low. Yeah.
I noticed that in your gene analysis, genes that are involved in oxidation and reduction seem to be favored during domestication and improvement. Any reason for that in your mind?
I don't know. It could be due to the, yeah, maybe you can provide a better examination. I mean, yeah, I don't know.
It was very striking in contrast to the disease genes were being unfavorable, so
Yeah.
they were being reduced. That makes a lot of sense. But why oxidation reduction genes involved in those pathways are being favored? It's interesting.
Yeah.
Yeah, maybe a weird question. So, commercialization of gene names or genome names, Heinz, is that a common thing, or?
I'm sorry.
Like, so
The carnival name.
the Heinz genes, like Heinz ketchup.
Uh-huh.
And is that a common thing? I think you could not do that in C. elegans. So, is that kind of a commercialization, like that the companies
Oh, you
get their own gene names?
You mean why we use Heinz?
So about the name itself, isn't that weird?
No, the carnival name is Heinz 1706.
Okay.
So you mean why we choose that one, or why we name that?
No. Isn't it related to the Heinz ketchup company?
Yeah, it is.
And is that a common thing? I'm just wondering.
No, I mean, so
Can we sell our genes to companies? When we have like
there's a history here.
Google Gene 1 or
So when we initiated this tomato genome sequencing project, actually at that time that was still Tansley. And we want to sequence the tomato genome. At that time we used back-to-back sequencing strategy. And we needed to have a back library, right? That Rod Bates group from the University of Arizona, at that time, I mean maybe a couple of years ago, he planned to construct a back library for tomato. He sent emails to companies, to consortium. I think only Heinz replied, see, I can provide seeds with Heinz seeds to you. And then he constructed the back library using the Heinz cultivar. Then later, when we do the genome sequencing project, we have to stick with that. We don't want to construct another back library. To be honest, Heinz is not a good reference, good cultivar for reference, especially for research. I mean, before Heinz genome published, no one used Heinz in their lab. Maybe as a Craig and maybe too. But I mean, because of this history, they used Heinz as a reference. And it happens, and also the name of the company.
Maybe we can give Fei one more round of applause.